Okay, so
Where is everyone?
Mary sleeping. Yeah, that's suspense today. Is anything going on? Last night was
Halloween.
not for.
Too much candy.
Yeah. Okay, so today first we'll quickly finish the rest few slides of the digestive system, and then we'll start the metabolism. Okay, so we are talking about the liver. Liver is the largest. There you go. Oh, he was here.
Yeah.
Okay. So liver is the largest gland in our body, and liver has 4 lobes: right lobe, left lobe, quadrate lobe, and caudate lobe. We have talked about that. Which one is the largest lobe? The right lobe, right? That's why we say that liver is located in the right side, because right lobe is huge and located in the right side. So if you see posterior inferior view, that means below the liver, little bit behind, this is the inferior view of the liver, and you can see all those 4 lobes from bottom. Now you see an area, an indentation through which the hepatic artery and hepatic portal vein enters into the liver. That indented area or indentation is called porta hepatis. Okay, so porta hepatis, like the hilum of the lung. You remember the hilum of the lung? Yeah. So similarly, in the liver, the indentation in the inferior surface of the liver, where the hepatic artery and hepatic portal vein enter into the liver, that is called porta hepatis. You just remember that.
How do you spell that?
It is there. See, left side in the middle. Porta hepatis. Yeah. Okay. Now, another thing you need to remember, the pathway of bile.
What?
You know that the pathway of bile. How bile reaches from the liver to the duodenum. We have talked about that in last class, right? You know that where the bile is produced? In the liver. And where the bile is stored? The gallbladder. Where the bile is drained? Into the duodenum. Very good. So after the bile is produced in the liver, how the bile reaches to the duodenum, that pathway. From the right and left lobes of liver, 2 ducts come out. Those are called right and left hepatic ducts. Okay? And then they join together and form what? The common hepatic duct. And common hepatic duct meets with cystic duct. Cystic duct comes from where? Gallbladder. So from the gallbladder, the cystic duct comes and meet with the common hepatic duct and form the bile duct. Then just at the entrance of bile duct into the duodenum, the main pancreatic duct meets. Where? At the ampulla. You remember that? And then enters into the duodenum, and that opening in the duodenum is called the major duodenal papilla. So again, I am showing, although I showed you in last class, but I am just quickly repeating here. The passage of bile. This is the left lobe. This is the right lobe. So right hepatic duct, left hepatic duct, they meet and form the common hepatic duct, and then from the gallbladder, the cystic duct comes out, and that meets with common hepatic duct and form the bile duct. And you see bile duct is very long and going all the way down to the duodenum. Just before it enters into the duodenum, it meets with the main pancreatic duct, and that area where they meet is a little bit dilated, expanded, wider. That part is called the hepatopancreatic ampulla. And then the opening inside the duodenum, that is called major duodenal papilla. Okay? And from the main pancreatic duct, a branch goes to the upper part of the duodenum, above the major duodenal papilla, and that opening is the minor duodenal papilla. Okay?
So some of the bile comes from the liver, and some of the bile comes from the gallbladder.
Yes. Most of the bile comes from gallbladder, because when we need bile, you eat a lot of fatty food, you need more bile, right? So that will stimulate the gallbladder to contract, and contraction of gallbladder will release plenty of bile inside the duodenum. Okay. Now, if you see the microscopic structure of the liver, if you cut the liver and see under the microscope, you will see that liver is formed by lobules. Many lobules, they join to each other inside the liver. And what are the lobules? Lobules are hexagonal structure. They have hexagonal structures, that means they have 6 angles, and the lobules are called the functional unit of liver. Okay? Those are the functional units, and the lobules perform following tasks: process nutrient-rich blood, and lobules composed of hepatocytes. Also, inside the lobule, you will find the Cooper cells and bile canal equivalent. So, the liver consists of lobules, and lobules are hexagonal structures, that means 6 angles. And if you see a lobule, the liver cells are called hepatocytes. Hepatocytes are arranged very nicely like this. From the center, they form chain like this and move towards the boundary. Okay, so this is the lobule, and inside the lobule, the hepatocytes or liver cells are arranged like this. From the center, they radiates towards the boundary like this. Okay, so that's the structure of lobule, and hepatocytes are the liver cells. Now, inside the lobule, you will also see phagocytic cells, and those phagocytic cells, kind of macrophages, they engulf the microorganisms or other dead cells. Those phagocytic cells are called Cooper cells. I may ask you that what is the name of phagocytic cells or macrophage type cells inside the liver? Those are called Cooper cells. Cooper cells are nothing but macrophages inside the liver. Okay, they have different names. But main cells are hepatocytes. Okay. So you see, if you just see the liver tissue under the microscope, many lobules, and lobules are hexagonal, and inside the lobule there is a central vein. That means a vein is located in the center of the liver here. Okay?
A vein inside each cell.
Each lobule. Each lobule. This is just one lobule, okay? This is one lobule, this is another lobule. So in the center of the lobule, there is central vein. And lobules are filled with what? Cells, hepatocytes. Make sense? So lobules are filled with hepatocytes. And you see how the hepatocytes are present inside the lobule like this. So all these are hepatocytes. And also inside the lobule there are few large phagocytic cells. Those are macrophages. Okay. Now, if you see this picture, again you see a lobule. This is a lobule, and this is the center of the lobule. So this is the central vein, okay, passing through the center of the lobule. And you see how I have drawn there, very similar. From the center of the lobule, the hepatocytes are going like this, forming chain and going to the boundary. Okay. Now, these are the branches of the vein, right? These are the branches of the vein. And one thing here important, you just see that these are the hepatocytes. That means the main liver cells, and each hepatocyte is attached to those tiny canaliculi. So those are bile canaliculi. And liver cells produce and secrete bile. We know that. You remember I said that if I ask you where the bile is produced, in the liver, right? The liver cells produce and secrete bile. So that bile must be collected by the branches of bile duct. So you see these are the tiniest channels or canaliculi. They collect the bile from the hepatocyte. That's why each hepatocyte is attached to the bile canaliculi, because the bile should enter into the canaliculi and then they will enter into the bile duct. They will be drained into the bile duct.
You said the bile canaliculi collects bile from what?
Collect the bile from the hepatocytes. Okay. So that's why each hepatocyte has attached bile. Okay, canaliculi, bile canaliculi. So cell can directly release the bile into the canaliculi. Then canaliculi will join together and drain the bile into the duct, larger duct. Okay, so it's like small tubules, and they are draining into larger tubules. So now another thing you need to remember here, in each corner of lobule you have three structures in each corner of the lobule. How many corners the lobule has? Six. Okay. Which three structures? A vein, an artery, and a bile duct. Those three structures are present in each corner of the lobule. Okay.
What do you have? You have a vein
And
an artery.
And a bile duct. You see here, bile duct, portal venule, that's the small vein, and portal arteriole. And these three structures together is called portal triad. So if I ask you what is portal triad, and where the portal triad is located, portal triad is located where? At each corner of what? Hepatic lobule, right? Lobule. And portal triad consists of what? Three structures: bile duct, arteriole, and venule. The arteriole is the small artery, and venule is the small venule. Yes.
Oh, so the middle, the central vein only has a vein. It doesn't have an
No,
artery.
only vein. Yeah, only vein. Okay.
So isn't there usually an artery wherever there's a vein?
Yeah, in case of liver, you know that hepatic portal circulation. What's the function of liver? Main function, if I ask you, number one, metabolism. Metabolism, right? And from the intestine, hepatic portal circulation, hepatic portal vein will bring the nutrient-rich blood into the liver, right? So the vein will deliver the nutrients to the liver cells where the metabolism will occur, right? So that's why you see the veins are everywhere inside the liver and ducts are everywhere, the canaliculi. Those two are important tasks, right? One is the duct is produced by the liver, bile is produced by the liver that should be released into the duct. Number two, the nutrient comes from the intestine and that should be given to the liver cells. Make sense? The nutrients should be given to the liver cells for metabolism. That's why you see extensive veins and branches of ducts.
The nutrient goes through the blood or it goes in through the intestine? Where from the intestine do the nutrients go into the liver?
That we have talked hepatic portal circulation from the intestine. The nutrient-rich blood should go to the liver, okay? And the vein will bring that.
So the blood brings the nutrients, it doesn't get the nutrients directly from the intestine?
No, blood will take that nutrient from the intestine and deliver to the liver.
So it goes into the blood, the nutrients from the intestine, and then in the circulation it goes to the right.
Yes, liver, yes. Okay, so the functions of hepatocytes. Hepatocytes are the cells of the liver. Process blood-borne nutrients. As I just mentioned, that nutrients should be processed for metabolism in the liver cells. Store fat-soluble vitamins. So the liver cells store vitamins. Perform detoxification, another very important function of the liver. The drug, we almost, regularly we take different types of medicine or drugs, right? And most of the drugs have harmful or toxic elements. And where those toxic elements are neutralized in the liver. That's why you'll see sometimes, most of the time, you'll see if someone has liver disease, it says on the medicine that don't take this medicine if you have problem in the liver, because the detoxification of drug occurs in the liver. If your liver has problem, it will not be able to detoxify the toxic substance of the drug. So it will cause harm in your body. So detoxification of chemicals. Production of bile. We already know that liver cells produce and secrete bile.
Do we need to know that number?
Second?
Do we need to know that number?
900 milliliter, not necessarily, just know the functions, okay, important functions. Now we'll talk about bile. Bile is yellow-green alkaline solution containing two things, bile salts and bilirubin. Okay, now what's the color of bile? Yellow-green. And why the bile is yellow? Because of bilirubin. Bilirubin is yellow pigment. Okay? And that gives the bile yellow color. Now bile also has bile salts. And bile salts are very important for the fat emulsification and absorption. Two things. So bile salts are important for fat emulsification and absorption. What is that? If this is your intestine, okay, and these are the fat globules. You already know that fat molecules are larger or smaller? Big. Very good. So bile does two things. Bile salts, after the bile is released into the duodenum, bile salts will get attached to the outer surface of the fat globules or large fat molecules, okay, like this. And will reduce the surface tension of the fat globules, and that will, when the intestine will move, that will break the large fat globules easily. So the bile salts will get attached to the outer surface of the fat globules and reduce the surface tension. So a small movement will break the fat globules into smaller molecules. Okay? It's like, you know, when you wash your clothes, what you put what?
Detergent.
Detergent.
It's a simple-
Bile just exactly work like that. Okay? What your detergents do, they get attached to the dirt particles, outer surface of dirt particles, and break them. Okay? So bile salts will do exactly the same. Will get attached to the outer surface and will break the large fat globules into smaller structures. Now, the fat globules become smaller. Now what happens? Again, the bile salts will get attached to the outer surface of those smaller fat globules and do what? Cover outer surface like this. We know that for fat it's difficult to pass through the cell membrane because cell membrane is phospholipid bilayer. Do you remember that cell membrane structure? Phospholipid bilayer. That means it is mainly lipid structure, right? So what will happen that the bile salt, just know that bile salt will cover the lipid surface of lipid globules, so will make it more water soluble. Because bile salts are water soluble, so will make the fat water soluble. So
Why does that make it more water soluble?
so it will easily pass through the wall of the intestine and enter into the blood. Enter into the blood. That is the absorption, right? So we'll do bile salt will do two things. Number one, emulsification will reduce the surface tension and break the large fat globules into what? Smaller. Okay, number one. And number two, it will again cover the smaller fat globules and make it what? Water soluble. So it will easily pass through the wall of the intestine and enter into the blood. That is the absorption, helps in absorption. Just know those are the two important functions the bile salt performs. Now, the gallbladder. Gallbladder is a muscular sac that stores and concentrates bile by absorbing its water and ions. So, gallbladder performs two functions. One is storage of bile. Number two, increase the concentration, concentrates the bile. How? If this is the gallbladder, bile is here. So from the bile, the water and ions will be absorbed. So if you take the water out from the bile, the concentration of bile will increase. Okay, so that is another thing will happen inside the gallbladder.
Explain that again.
Okay, so you understand that gallbladder stores bile, right? Okay. Number two, when the bile is sitting inside the gallbladder, the water part of bile will be taken back into the blood through the wall of the gallbladder. So from here, the water will enter into the blood vessels of the wall of the gallbladder. Make sense? So if water is taken back into the blood, then the concentration of bile will increase, right? From any solution, if you take the water out, then the concentration will increase. So that way. So if I ask you the question like this, that if you take a drop of bile from the liver and take a drop of bile from the gallbladder, which one will be more concentrated? Many times, 100 times concentrated. Okay? The reason is the water is taken out from the bile in the gallbladder.
Well, when it goes into the duodenum, some of the bile comes from the liver and some of the bile comes from the gallbladder.
Yes, but most of them come from, most of it comes from
From the
gallbladder.
gallbladder.
Yes.
So if a person doesn't have their gallbladder,
Then you have to do two things. If the gallbladder is removed, two things you have to do. Eat less fat, because bile is important for the digestion of fat, right? So you will eat less fat food. Number two, you have to take supplementary bile, supplement from outside, okay? That will help in
so the liver won't give enough.
Right. Liver is still producing, but not highly concentrated. And liver doesn't know when you need more or less. But your gallbladder knows, because I told you, when you eat a lot of fat, that sends signal to the gallbladder, and gallbladder will contract and will, release the bile. But liver, you cannot tell the liver, because liver doesn't store bile, right? So it cannot release plenty of bile if you need. Suddenly.
If the gallbladder is missing and the liver keeps producing bile,
Bile is slowly going to the,
so it won't have anywhere to put it.
right. Okay, large intestine. If you see the large intestine from outside, two structures are clearly seen. One is called Tinea coli and another is house trap. Tinea coli is formed by three bands of longitudinal smooth muscle in the muscularis. What is that? If this is the large intestine, you will see band of smooth muscle fibers like this attached to the large intestine, and that band of smooth muscle fibers is called Tinea coli. Inside the Tinea coli, there are three longitudinal bands are present, and haustra are pouches. If you see the large intestine from outside, you will see it's not a straight tubule or tube. It has many pouches attached to each other and form the large intestine, and each pouch is called haustra. So those are pocket-like sacs. So if you see here, this is the picture of a large intestine, and this is the Tinea coli, thick band attached to the large intestine, Tinea coli here, and these are the pouches. So not a straight tube, it's like many pockets are attached to each other, and each of these pocket-like structure or sacs or pouches, whatever you say, is haustra. And haustra is more than one, and haustra is one. If I say only one, that's haustrum, and if I say more than one, then haustra. Okay, now we'll see the parts of the large intestine. The first part is the cecum. This is the starting of the large intestine, cecum, and from the cecum, a finger-like, tiny finger-like structure or worm-like structure gets out. That is called vermiform appendix. Okay?
And what is the purpose of the appendix?
Nothing. Appendix doesn't have any purpose, any function. Okay? It's useless.
So it doesn't do the job of the large appendix?
No, it doesn't do
Only the
anything.
large,
It creates problem. Otherwise, it doesn't do anything. Okay. So, vermiform appendix is a small finger-like or worm-like extension from the cecum. And after the cecum, next part is the ascending colon. And then ascending colon suddenly turns in right angle and becomes transverse colon. And from the right side, it goes all the way to the left side. Okay? And then suddenly start to go down, and that's the descending colon. And then next part is the sigmoid colon, and then rectum, anal canal, and anus. So those are the parts of large intestine. Now, where the ascending colon becomes the transverse colon, that angle is called right colic flexure, also called hepatic flexure, because you know that liver is located here. Liver is here, so this part is very close to the liver. So that's the hepatic means liver.
So you said
colic
that's where the ascending turns to transverse,
Yes.
right?
Yeah, just this area is below the liver. So that's the hepatic flexure. And then this angle, left colic flexure, is attached to the spleen. That's why that is also called splenic flexure, because it is attached to the spleen. And now, vermiform appendix. I told you that vermiform appendix has no significance. It is actually an anomaly, developmental anomaly that doesn't perform any important function in our body. Okay? Vermiform appendix is filled with, if you see the wall of vermiform appendix, it is filled with lymphoid follicles. But you already know that lymphoid follicles are present in the wall of the intestine. You remember the Pearce patches? And the small intestine, large intestine, so long, right? Tube. Compared to that, appendix is very small, and that small amount of lymphoid follicles is not really, necessary for our body, because we have lymphoid follicles not only in the intestine, we have lymphoid follicles in all lymphoid organs, right? In the
Don't they
spleen,
think the
in
appendix used to
the
have a
tonsils,
function?
yes.
Don't they think the appendix used to have a function? I've heard that before.
What's that one?
I don't know.
Okay. So it doesn't have any function. It's a functional animal. But it creates problem, as I told you, that what problem? You all know that. Appendicitis, right? That can sometimes be dangerous for your life if the appendicitis occurs, and if you don't remove on time, that will swell up, will get bigger and burst. Now the problem is, when the appendix will burst, then you know that inside the large intestine, after the absorption of nutrients, the unnecessary, useless foodstuffs are there, right? And those are very poisonous. And if the appendix bursts, what will happen? The foodstuff will enter into the peritoneal cavity, and peritoneal cavity is filled with what? Peritoneal fluid. And once that poisonous fluid will enter into the peritoneal cavity, into the peritoneal fluid will take it everywhere. And inside your abdominal cavity of important organs, right? So they will easily go and infect the important organs and will kill you. Okay? So that's why when appendicitis is detected, immediately removal is important, right? Remove the appendix.
Well, how painful does it
So
have
when
to
it
be
breaks,
in that area of the body before, before you
Just say again.
I said when it bursts, how do they control it? Because one of my nephews burst about like 3-4 months ago.
Yeah, if
And
that-
he actually drove to the hospital.
Yeah.
While it
he's
just
okay?
burst. He's all right.
Yeah, immediately after, it takes some time to spread and infect other organs, right? So if you wash and give antibiotic, that will. But if you wait, that could be that can lead to shock, and the person may die because of poison spreading throughout the abdominal cavity. Okay. Now, why appendicitis is common? Appendicitis you very often hear. The reason is end artery. You know, in some areas in our body we have end arteries. What does it mean? End artery means there is no anastomosis.
Right.
Okay? And in end arteries, it is very easy to get block like this. And when that happens, see here, if there are anastomoses, then what can happen? That area can get blood through, you know, you remember in the heart, through another channel, right? But if end artery and blockage occurs here, this area will die and will, because inside the appendix we have the food stuff, so this dead tissue will easily get infected and will cause infection. Okay? Okay. Here, just remember two things. The connective tissue membrane that is attached to the transverse colon, that is called transverse mesocolon. So it's like just mesentery of transverse colon, transverse mesocolon. And the connective tissue membrane that is attached to the sigmoid colon, that is called sigmoid mesocolon. And they are very similar to mesentery, you know, that they connect the organ to the wall of the abdomen. Just to hold the structure in right positions. Just remember those two mesocolons. One is transverse, another is sigmoid mesocolon. Rectum and anal canal. So, if you see inside the rectum, there are 3 valves present inside the rectum, and those valves stop feces from being passed with gas. So 3 valves are present inside the rectum. Inside the anal canal, that's the last part of large intestine, anal canal, and there are 2 sphincters in anal canal. One is internal anal sphincter, another is external anal sphincter. And this is important. Remember that external wall, the external sphincter is formed by skeletal muscle, and internal one is formed by a type of muscle, smooth muscle. So now, if I ask you which one you can control,
So the external.
skeletal or smooth?
The external.
External, because we know that we can control the skeletal muscle, not the smooth muscle, right? The skeletal is voluntary. So, the external one is the skeletal muscle, remember that. And what happens, let me see if there is any picture here. They didn't show the internal sphincter, so let me draw here. If this is the anal canal, there are 2 sphincters. One is here and another is here. Okay, so this is the internal and this is the external, and this is skeletal and this is what? Smooth. And this is voluntary, this is involuntary, right? Now, when we decide to do defecation, what we do, a signal from the external sphincter, we can easily relax the external sphincter voluntarily. So we do that. When we relax the external sphincter voluntarily, the signal goes from the external one to the spinal cord. Spinal cord. Okay? So from the external sphincter, when we decide voluntarily to relax, when we relax that, a signal, nerve signal, goes to the spinal cord. And then the spinal cord will send a signal back to the internal sphincter, which is smooth muscle. And we do what? Relax the internal sphincter. So when the internal sphincter will be relaxed, then the feces will easily move down. Make sense? So both sphincters are now relaxed. If you only relax the external, internal is still closed, then the stuff will not move down. Okay? So you have to relax both. But which one you relax first? External, because we can't control it.
So
And then that will send signal where? To the spinal cord. And the spinal cord will send signal to the internal to relax, because we cannot do that, right? Our nervous system will do that.
So what, when a person feels the need to go
Yeah.
to the bathroom,
Yeah.
so,
First you relax the external one.
but nothing has, it's, nothing has been relaxed, not the internal or the external, it's just that there's a sense of it being there, or what?
First,
What causes a person to feel the need to go?
the pressure. The pressure that you on the wall of your large intestine.
So babies, they don't have,
Yeah.
they haven't learned to have control.
Not only learning, learning is one part. Another part is in small kids, probably you remember from NP1 that Babinski's side, the motor neurons are not developed very quickly in small infants. It takes time to develop the motor part of the brain. That's why they don't have any motor control. Motor control will control the skeletal muscles, right? So that's why they cannot control the sphincter. Okay, now we'll talk about the pancreas. Pancreas is a gland present horizontally in your abdominal cavity like this, horizontally, not vertical. And pancreas is what type of gland? Is a mixed type gland. Okay? Why we say pancreas is a mixed type of gland? Because it has both endocrine and exocrine parts. Okay? So pancreas has both endocrine part and what? Exocrine part. That's why it is called mixed type gland. Now, endocrine part of pancreas is called pancreatic what? Pancreatic islets. Okay? So, the endocrine part of pancreas is called what? Pancreatic islets. Also called islets of Langerhans. I don't know if you have heard, but islets of Langerhans or pancreatic islets. Same thing. And exocrine part is called pancreatic asyni or assigner part of pancreas. Pancreatic asyni or assigner part of pancreas. So two parts, right? Now, the endocrine part will produce two very important hormones. What are those hormones? One is insulin and another is glucagon. So those two hormones are released by the endocrine part, that means from the pancreatic islets. And pancreatic asyni or assigner part produces and releases the pancreatic juice. We have talked about that before. So pancreatic juices are what? Proteolytic enzyme, lipolytic enzyme, and amylase. So they will break the protein, they will break the carbohydrate, they will break the lipids. So digestive enzymes, in one sense you can say, one word you can say, digestive enzymes. Okay, so proteolytic, lipolytic, and carbohydrate digestive enzymes. So you see that endocrine part of pancreas is not related to digestion at all, right? It is related to the regulation of what? Sugar, blood sugar. Insulin does what? Increases or decreases the blood sugar?
Decrease.
Yeah, that's why diabetic patient takes insulin to reduce the sugar, right, in the blood. And glucagon does just opposite. So what is the function of glucagon? Increases sugar. Okay, and both are released in our body, and the balance of glucagon and insulin will keep the blood sugar constant. If the blood sugar increases, which one should be increased more? Insulin. If our blood sugar is less, the glucagon will be released and will increase. Okay, so blood sugar is regulated by these two hormones, the endocrine part of the pancreas. And the pancreatic juices, you remember the main pancreatic duct? The pancreatic juice, the main pancreatic duct inside the pancreas, the main pancreatic duct. So that goes to the duodenum, enters into the duodenum. Yeah. So the pancreatic juice will be released into the main pancreatic duct. That will take it to the where? Duodenum. Duodenum. You remember the?
We're a little lost.
Okay. So do you remember the picture of pancreas like this? And there was a duct like this, and accessory pancreatic duct. Do you remember that? Where the main pancreatic duct goes? Where it goes? Do you remember that?
Yeah.
Major duodenal papilla.
That's one.
And the bile duct comes down. Do you remember? This is the ampulla, hepatopancreatic ampulla. So I'm talking about this. Okay. So the pancreatic juice will be released into the main pancreatic duct here, and that juice will go where? Into the. Did you understand? Okay. And we know that it will digest, right? The proteins, carbohydrates, and fats. Just know the information I gave you. Okay. Okay, now we'll talk about metabolism. We'll talk about few things, not into detail, but the basic things about metabolism. First, we'll talk about the processes of metabolism. Metabolism indicates all chemical reactions in our body. You know that thousands of chemical reactions are continuously being taken place in our body, right? Inside the body. All chemical reactions together called metabolism. And metabolism or chemical reactions in our body can be divided into two types: anabolism and catabolism. So we'll talk about that. Then we'll talk about the metabolism of carbohydrates, the metabolism of fats, and metabolism of proteins. So we'll talk about those organic compounds. You know that what are the organic compounds? Organic compounds are proteins, carbohydrates and fats, right? Those are organic compounds. Now if I ask you, is water an organic compound? Water is not. Acids, bases, most of them, those are not organic, right? So organic are what? Proteins, carbohydrates, fats, and nucleotides. Those are, let me write it down here. So organic compounds, we'll talk about the organic compounds. What are the organic compounds? Proteins, carbohydrates, fats or lipids, and nucleotides. Okay, nucleotides. So those are the organic compounds. If I ask you what are the organic, those are the organic compounds. So we'll talk about the proteins, carbohydrates, and fats.
Nucleotides?
Yeah, nucleotides. And then we'll talk a little bit about the vitamins
Where do nucleotides come from?
inside the cells.
It comes from the food we eat? No.
Inside the body, we have the organic compound. So, as I mentioned, first thing, if I ask you what is metabolism, right? In simplest way to say is all chemical reactions in our body together is called metabolism. And now, metabolism has two branches, anabolism and catabolism. Catabolism, if I ask you what is catabolism, catabolism is breakdown processes. So breakdown. Catabolism does what? Breakdown. And anabolism is synthesis. That means production. Just opposite. If something new is produced, that is anabolism. If something is destroyed, that is catabolism. Okay? So again, you see, catabolism is the breakdown of organic substances, and anabolism is the synthesis of new organic molecules. And some chemical reactions in our body will produce new things. That's the anabolism. Some chemical reactions will break the proteins, carbohydrates, fats, right? Those are catabolism, catabolic reactions. Okay, now the catabolism is essential in our body for what? To produce energy. So catabolism produces ATPs, and we know that energy comes from ATP. So if I ask you which metabolic process will produce ATP, catabolism or anabolism? Catabolism. Why? The catabolism, we know that breaks the proteins, carbohydrates, fats, right? And our energy comes from the proteins, carbohydrates, and fats. Okay. Now, we'll talk about how the nutrients are processed in our body. Very simplest way, we'll talk about that. You eat food, right? Yeah. And what happens after you eat the food? First thing is digestion, right? Breakdown. It starts in your mouth, and as it goes down, further and further breakdown occurs, right? That's the digestion. So, first process is digestion in our body, and then when the food enters into the intestine, the breakdown process is almost done, and the nutrients will be taken into where? Into the blood. And that process is called what? Absorption. The process by which the nutrients are taken from the intestine to the blood. So next is absorption of nutrients. And then the blood will take the nutrients where? I have just mentioned, mainly to the liver. You remember I said that the nutrients should go to the liver, as well as blood. Once the nutrients will enter into the blood goes where? Everywhere in the body, right? So most of the nutrients will go to the liver because liver is the main site for metabolism. You remember I said that if I ask you tell me one function of liver, that's the metabolism, right? So liver is the main organ for metabolism, but metabolism occurs everywhere in the body, remember. So most of the nutrients from the intestine will be taken to the liver, but also will be given to all other tissues in our body. Okay, so that is the transport of nutrients. So first digestion, then absorption, then transportation of nutrients. Then what will happen? After the cells of your tissue receive the nutrients, nutrients enter into the cell and two things happen. If this is the cell, okay, and nutrients will enter into the cell, and inside the cell we have mitochondria, right, and cytoplasm here. So first the nutrients will enter into the cytoplasm part, the fluid part, and enter into the cytoplasm, and both anabolism and catabolism can occur in the cytoplasm.
The cytoplasm is the blood?
Now, cytoplasm is the fluid inside the cell. You know that, right? Cytoplasm. Okay. So, when the blood will deliver the nutrients into the cell, first in the cytoplasm part, the fluid part inside the cell, inside the fluid or cytoplasm, anabolism can occur or catabolism can occur. So, for example, glucose. Is entering into the cell, and catabolism will do what? Will break the glucose, right? Catabolism is the breakdown process. So, glucose will be broken down, and that is called glycolysis. Glycolysis. So, glycolysis is the breakdown of glucose. Breakdown for what? Why the breakdown should occur? To produce ATP. We know that, to produce energy. So, that catabolism, glycolysis, will produce ATP. Now, if our body, remember that, if our body needs ATP or energy, then the breakdown will occur, right? But if your body does not need enough ATP, but you are taking lot of glucose or food, then what should happen? Breakdown should not occur, right? Because our body doesn't need ATP, that many ATP. So, in that case, what will happen? Glucose will not be broken down. Glucose will be converted to glycogen. That is the stored form of carbohydrate or glucose. Okay? So, you see here, the glucose can go into two pathways. One is, if your body needs ATP, then glucose, what will happen? Catabolism will occur, right? Breakdown of glucose will occur. And that will produce ATP. Is it clear? Mm-hmm. If your body, if you have taken a lot of glucose, your body doesn't need that many, then what your body will do? Body will store it, right? For future use. So, that storage form of glucose is called glycogen. Now we see that glucose, from the glucose, new molecule is being synthesized or produced, and that process is anabolism. So this is catabolism, glycolysis, and synthesis of glycogen is anabolism. Production of new things, not breakdown, but producing a new molecule. Okay.
Where is it stored?
Inside the cells. Inside the cells of the body. Now, first glycolysis will occur in the cytoplasm. Then what will happen? The chemicals will enter into the mitochondria, and inside the mitochondria, the last part of glycolysis will occur. Further breakdown will occur, and that will produce carbon dioxide, water, and further ATPs. So more ATPs will be produced in the mitochondria. So first part of glycolysis will occur in the cytoplasm, and last part of glycolysis will occur inside the what? Mitochondria. Okay? And in the cytoplasm, only few ATPs will be produced. That means in the first part of glycolysis, only few ATPs will be produced, and inside the mitochondria, most of the ATPs will be produced inside the mitochondria. Okay? That means in the last part of glycolysis, we will get most of the ATPs. Okay, now why metabolism is essential? Metabolism is essential for metabolic turnover, growth and cell division, and special processes such as secretion, contraction, and propagation of action potential. First, metabolic turnover. What is that? You know that continuously some cells inside the body are dying or being destroyed. Do you know that? Like red blood cells. Just simplest way thing that we know that red blood cells lifespan is 3 to 4 months, right? So red blood cells are continuously dying. All other cells, your skin cells are dying. Inside the body, when you get infection or any friction, the cells are dying. Inside your mouth, the cells are dying, right? When you crash the food. So continuously, some cells are being dying. And now, to replace those dead cells, our body needs to do what? To produce new cells, right? And that is the turnover, okay? To replace the dead or injured cells. Not only complete death, think that when you get infection inside your body, cell may not die completely, but the wall of the cell membrane can be destroyed partially, right? Then we need to fix that. So new protein should be produced and new fat should be produced to fix that wall of the cell membrane. So those things are continuously taken place.
Are these things that sleep, that you need sleep for, that happens a lot at night?
Yes, when your body is in resting condition, that will happen more. Okay. Growth and cell division. So when our body grows, more cells should be produced, right? And to produce from 1 cell, 2 cells, then from 2 cells, 4 cells, and 4 to 8 to 60, right? That's how the cell multiplication occurs and body grows. Now, to produce new cell, you need new protein, new carbohydrate, new fat structures, right? So, growth and cell division. For growth and cell division, we need the synthesis of new organic compounds. Make sense? Synthesis, production. That's why we eat food, and from those food, new proteins or new fats will be produced to grow the body. Special processes. Why we need metabolism for special processes? We need metabolism for special processes because special processes need energy. Very simple. Let me give you an example. Muscle contraction, right? When muscle contraction occurs, the muscle fiber should get shorter, right? And you remember actin, myosin, attachment, and power stroke, like this, we need ATP. For muscle contraction, we need ATP, right? Energy. For secretion, you know that if this is the secretory cell, the vesicles contain the chemicals. Those should be released. So, see here, few things should be done before the secretion. The vesicles These are glandular, this is glandular cell. The vesicles contain the chemicals. Those vesicles should be taken to the apical end of the cell. So someone should take it there, right, to the end of the cell. And then what should happen? The rupture of the vesicles should occur, right, to release. So to take it there, to rupture the cell membrane, everywhere we need energy, right? So continuously energy is being used for those special processes, secretion, contraction, and preparation of action potential. That's why metabolism is essential. Okay, now the production of ATP. ATPs are produced at two levels. What are those? First, substrate level phosphorylation. And substrate level phosphorylation includes glycolysis and Krebs cycle. So those two are substrate level phosphorylation. And then oxidative phosphorylation, that occurs in the electron transport system of the mitochondria, inside the electron transport system of mitochondria. So that is called oxidative phosphorylation. So we'll
What's
see.
the Krebs cycle?
Second,
What's the Krebs cycle?
Krebs cycle is a chain of reactions. You know cycle means what, right? Cycle is going like this, right? So most of the reactions, chemical reactions in our body goes like this, from one to another, right? But in Krebs cycle, what happens when the chemical reaction occurs here, that will trigger another reaction, and that will trigger another, and will go like this, and will come back to here. So it's a cyclical form of chemical reactions, chain cycle of reactions. Okay, Krebs cycle has another name that is called Krebs cycle has another name, TCA, right? Tricyclic acid cycle, TCA, or tricyclic acid cycle. Anyway, so say here, I am showing you here. So the ATP synthesis occurs in two mechanisms or two phases. One is what? Substrate level, right? And another is oxidative. I just have talked about that, those two. So now we'll see substrate level. This is the cytoplasm inside the cell. That means, you remember I talked about when the nutrients will enter into the cell. First, where the reactions will occur? In the cytoplasm. Do you remember, everybody? In the cytoplasm. Then the last part will occur where? Inside the mitochondria. Okay? So remember that. So after you eat proteins, carbohydrates, fats, what happens? Digestion occurs, right? After you eat the food, proteins, carbohydrates, fats, first thing happens is digestion. And digestion will do what? Break the food. So protein digestion will produce amino acids, because we know that amino acids join together and form the protein chains. Okay? So breakdown of protein or digestion of protein will produce amino acids. Digestion of carbohydrates will produce glucose and other sugars like sucrose or lactose, right? Those will be produced. And digestion of fats will produce two things, glycerol and fatty acids. Okay? So after digestion of proteins, you will get amino acids. After digestion of carbohydrates, you will get the glucose and other sugars. And after the digestion of fats, you will get glycerol and fatty acids. And now see the difference between food and nutrients. We often use those terms, right? Food and nutrients. What's the difference? If I ask you, what are these? These are food. These are food, right? And these are nutrient molecules. So food becomes nutrient molecules after digestion or by digestion. Okay? And nutrients are very small molecules, so they can enter into the blood, right? And then blood will do what? Transport the nutrients to the cells. Because our cells, tissue and cells need nutrients, right? So the nutrients will enter inside the cell. This part, whole part is the cell. So now, if I ask you, the nutrients will first enter into which part of the cell? Cytoplasm of the cell, right? So this is the cytoplasm of the cell, and this part is the mitochondria of the cell. Okay? So the first part of glycolysis, as I mentioned, will occur in the cytoplasm, right? So glycolysis will occur in the cytoplasm, first part, and in that part of glycolysis, what will happen? The glucose will be converted to pyruvic acid. Very important information. In the cytoplasm, the glucose molecule will be converted to pyruvic acid by glycolysis. Glycolysis is breakdown of glucose, right? So one molecule of glucose will produce 2 molecules of pyruvic acid. So each glucose will produce how many pyruvic acid? 2. Okay? So remember that.
Could you go over that one more time?
In the cytoplasm, the first part of glycolysis will occur, right? And that will do what? Convert the glucose into pyruvic acid. You see that picture. Glucose is converting to pyruvic acid. And one molecule of glucose will produce 2 pyruvic acids. Okay? Is it clear? Okay. Then pyruvic acid will be converted to acetyl-CoA before it enters into the mitochondria. Okay? So the pyruvic acid will be converted to acetyl-CoA, and acetyl-CoA will enter into the mitochondria. And inside the mitochondria, 2 things will happen. So now here, there is acetyl-CoA is entering into the mitochondria, and last phases of glycolysis will occur inside the mitochondria. Now, inside the mitochondria, 2 things will happen. First, the Krebs cycle, the cyclic chemical reactions will take place. That is called what? Krebs cycle, or TCA cycle, that I have just shown you here. Okay, number of reactions, but they will go in cycles. And then the last part.
The Krebs cycle is that before it goes into the mitochondria?
No, this is mitochondria.
Okay.
This is mitochondria. Okay, so inside the mitochondria, I said how many things will happen? 2. First is the Krebs cycle, and last is the tricyclic acid, sorry, not tricyclic, oxidative phosphorylation. Okay, so 2 things will happen in the mitochondria. I'm repeating again. First is Krebs cycle or tricyclic acid cycle, and last is the oxidative phosphorylation. Yes.
What did you say the pyruvic acid is? The acetol?
Acetyl-CoA, acetyl-CoA, acetyl-CoA. Okay. Just no acetyl-CoA. Okay. So that will happen where? In the cytoplasm, right? And then acetyl-CoA will enter into the mitochondria. And inside the mitochondria, how many things will happen? 2. First is the Krebs cycle, and second is the oxidative phosphorylation. Is it clear? Okay. Now, I told you before, where you will get more ATPs in the cytoplasm or in the mitochondria?
Mitochondria.
Very good, you remember. So most of the ATPs we get inside the mitochondria. That means by Krebs cycle and oxidative phosphorylation. Only few ATPs we get in the cytoplasm, glycolysis in the cytoplasm. Now, one molecule of glucose normally produces 36 ATPs. So one molecule of glucose will produce how many ATPs? 36, okay? And among those 36 ATPs, only 2 will be produced in the cytoplasm. Only how many? 2 will get from the glycolysis in the cytoplasm. And 34 ATPs we get from combined actions of Krebs cycle and oxidative phosphorylation. So 34, yeah.
34?
34, yes.
So you said you get 36.
Yes, 2 from the glycolysis, right? 2 from the in the cytoplasm we get how many? 2. And 34 we get from the
Oh, so it
mitochondria.
equals totally from one
Yes.
glucose.
Yeah. So now, here in the cytoplasm, actually 4 ATPs are produced. I'm not going into details, just this is the last thing. 4 ATPs are produced in the glycolysis of in the cytoplasm, and but I said how many we gain? 2. The reason is other 2s are used to start the process. It's like you in the winter morning you start your car, right? So you spend some gas, little bit although, to warm it up, to warm the engine or start the engine, right? So 4 are produced, but 2 are used to start the reactions. Okay? That's why the gain is
4. 4 are produced in the cellular,
Cytoplasm.
and 2. So really 4 are produced, but 2 are used.
Okay, so we'll stop here and start from here next class.